Assalamualaikum, I'm Erika, and in this video I'm going to talk about prey identification using DNA barcoding and identification of invasive species of peacock using mitochondria DNA barcoding in Talabat Lake, Terengganu. This research was conducted at a man-made freshwater Talabat Lake, which is known for the vast diversity of the aquatic life and fishes that play important role to the local people. Hence, introductions of invasive species into this lake will cause reduction of native species and might disrupt the food chain and causes threat to the biodiversity. Introductions of invasive species or alien species into water bodies can be happen either by accident or purposely. Example of alien species in Malaysia include red-tailed catfish, flowerhorn, suckermouth catfish, and our main topic, peacock bass. Peacock bass is a voracious predator fish with wide prey range, threatening the native fishes including endemic species, and it purposely introduced to Talabat Lake by the anglers. So the issue was about invasive species of peacock bass can exert a high predation on prey fish population, which leads to decreasing of small prey fish in Talabat Lake. To solve this problem, we must know species of the peacock bass so we could enhance our understanding of that species in order to guarantee the safety of biodiversity in Talabat Lake. Also, by knowing the species of the prey, we're capable to identify which species is the preferred one by the peacock bass. The aim is to study the origin and species of key class species. Second is to identify prey species of peacock bass. Third is to compare the method between DNA barcoding and mitochondria DNA barcoding to identify fish species. The method used to identify peacock species is by using mitochondria DNA barcoding. The process starts from sample collection. Second is DNA extraction. Third is PCR, which is using CO1, and D-loop region where CO1 using CO1 fish to forward and reverse primer, while D-loop using T-Pro2 and HN20 primer. Then this PCR result undergo DNA barcoding to obtain the sequence, where BLAST in NCBI is used to identify the species. Next method is using DNA barcoding to identify prey species, which also start from sample collection. Second is taxonomic classification. Third is preparation of barcoding sample. Fourth is the extraction of DNA. Fifth is amplification using CO1 gene, where the primer used to identify fish is CO1 fish to forward and reverse, while unidentified invertebrate by using LCO1490 and HCO2198 primer. Then the result from PCR undergo DNA sequencing, and the sequence obtained are analyzed using cluster W. Results show a total of 670 BP of CO1 and 857 BP of D-loop mitochondria DNA were obtained after deletion of low-quality nucleotide at the 5' and 3' ends. The BLAST search sequence match results show highest percentage identity with 3 C. ocularis sequence as shown in this slide. Even though C. tamensis obtained highest sequence similarity, which up to 99.84%, however, it only score 1127 maximum score, compared to C. ocularis sequence similarity, which obtained 99.68% and the highest score of 1147. For D-loop gene, it shows highest sequence similarity for C. ocularis 98.95% with maximum score of 1531 and 1530. Hence, majority of peacock species in Talabat Lake is coming from C. ocularis species. In this slide, picture A shows sample obtained from Talabat Lake, and picture B shows phylogenetic tree where Caicla species in Talabat Lake are closer to species Caicla ocularis. The graph below shows the stomach content after each month collection, where the highest vacuity coefficient was observed in December, indicating their breeding and spawning season. The lowest vacuity coefficient observed in January, 26.3%, revealed its feeding initiations after breeding period. While the picture A, B, and C show total stomach content for 4 months, where 51.8% contain empty stomach, 43.1% unidentified fish species, and 5.1% unidentified invertebrate species. And the table in this slide show the monthly variations of peacock bass dietary. This table shows that our trilineata has the highest percent identity, which is 99.99%, and with only 2.5% of this species found in total of peacock bass stomach, while the highest percentage of each prey species identified using DNA barcode were also recorded with a high value of 15.2% in P. fasciata with 99.07% similarity sequence. Therefore, by using mitochondria DNA barcoding, the result shows C. ocularius is the species that introduced in Telabak Lake with the highest percent identity is 99.72 and 99.68%. However, the origin of this species is unknown. And current study also matched C. temensis with 99.84% show the origin is at states of Brazil. This finding is crucial for investigations of the C. ocularius impact to the Telabak Lake, where local people experience gradual declines of small-sized fishes, while DNA barcoding show that the highest prey in peacock stomach is P. fasciata with 15.2% and followed by Piranga 10.1%. All prey species found in peacock stomach is native species that less concern to people as such species Macrobranchium, where M. lanchesteris is important to local people as food. So, to compare between these two methods, mitochondrial DNA barcoding able to identify up to 99.72% sequence similarity, much better compared to DNA barcoding that only identify percent identity up to 99.20% of the same species of peacock. However, DNA barcoding also show the highest sequence similarity for prey species, which is 99.99% for Arthriliniata, or also known as scissor-tail rasbora. So, which one of these two methods is better? So in my opinion, both of these methods is suitable to be used to identify fish species. However, based on this study, it shows that mitochondrial DNA barcoding show better high percent identity of similarity for peacock species identification. Therefore, this finding could help us to understand more of their impact to biodiversity in Telabak Lake, and we also able to identify which species is endangered to extinction. Other than that, invasive species might affect government effort on promoting the lake as recreational and aquaculture center. Lastly, I believe mitochondrial DNA barcoding is useful to identify invasive species and origin of peacock, bass, and the second best is DNA barcoding, as it also show promising result in identify the prey species. So these are the references that are used, and that's all from me. Thank you for listening.